Welcome to the Physiology Lab at Maryville College. I'm Macy and this is Matthew, and today we'll be demonstrating the techniques that Dr. Crain's Science 150 students will be using for examining nucleic acids in strawberries. Each Science 150 lab group will generate their own question and use the scientific method to examine this question. We will show you the basic techniques for isolating DNA, and you will then be designing an experiment based on your own question.
So here's the technique. The first step is to thaw and mash your strawberries. Now, 3 frozen strawberries will weigh about 30 grams. Now, the reason we're going with strawberries is because each cell has 7 individual chromosomes and is an octoploid, which gives each one 56 total. Because there's so much genetic material in each of these, it's a really great option for this particular experiment. Now, we tried a lot of other food items in the process of designing this lab. We went through 2 different kinds of peas, 3 different kinds of ground beef, liver, tuna, and spinach. This was by far the best.
Make sure you take your time mashing this up.
At least a minute. This is going to take a while, and the more time you spend doing this, the more genetic material you're going to get out of them.
So it's been about 3 minutes and we've got a good puree of our strawberries. Now we're going to create the DNA extraction fluid. First, we're going to add a tablespoon of liquid detergent. We're going to use Dawn, but others may work as well. This detergent disrupts the lipid bilayer of the membranes, so both the cell membranes and nuclear membranes are dissolved, releasing the nucleic acids from the cells. Next, we're going to add half a teaspoon of salt. This salt removes the histone proteins that are bound to the DNA, keeping it tightly packed. Lastly, Matthew's going to add half a cup, or about 118 milliliters, of cold water. The cold temperature ensures that the DNA stays intact, keeping the enzymes in the strawberries from breaking up the DNA. You may need to stir it for a while to get it mixed up very well.
Alright, now we will add the liquid we just made to the mashed strawberries, seal it, and gently mix for about 1 minute. When this is done, we're going to pour all through a coffee filter to take out any particulate and leave us with just, hopefully, genetic material. Okay, these are just regular coffee filters. Now what this process is going to do is filter out all the cellular debris, like the cell membranes, the cell walls, whole pieces of the strawberry left, and maybe even seeds. And it's going to leave behind what was left in the cytoplasm, which is going to contain, hopefully, nucleic acids. So this is going to take a while. You got to take your time with this and maybe even use more than one filter. But let it drip just like it is now. And then when it gets to the point where it's stopping, we're going to take it up and gently squeeze the coffee filter to take out any remaining fluid.
Now we've finished straining, and now we're going to add an equal volume of cold isopropyl alcohol. We're using 91%, but there may be others that work as well. The alcohol is nonpolar and it repels the negatively charged DNA, causing it to precipitate out into this white, stringy, gooey stuff that you will see. Now you need to do what Matthew's doing and pour it slowly down the side of the cup so that you don't mix up the strawberry stuff with the alcohol.
Do this right, you're going to form a whole new layer of alcohol that rests right on top of the strawberry liquid. And watch carefully as this starts to happen, because you'll start to see the DNA precipitate if you've done everything correctly. So we've completed the process, and this is the final result. Now, it's going to be your job to tweak either the technique we've shown you today or something with the strawberries to see if you can increase or alter the yield of nucleic acids.
So have fun and good luck.